Hello students, today we are going to discuss in detail on the process of gene therapy. Any of the gene therapy experiments can be divided into 5 different steps, that is selection of the gene of interest, that is the gene which is of in good condition what we are going to use a particular genetic disorder, then selection of the plasmid and selection of the vector, what type of vector we are going to use, whether it's a viral vector or non-viral vector, that we are going to decide. And further we are going to see the selection of method in which we are going to incorporate the viral or non-viral vector, it is in vivo gene therapy or ex vivo gene therapy, then particular techniques what we are going to follow up in this case. Selection of gene of interest. One of the most crucial step in gene therapy is selecting the gene of interest. So we have to select the appropriate gene of interest based upon the disease type. For example, the person is having a deficiency of dystrophin gene. In case of Duchenne muscular dystrophy, we have to choose only the good gene of dystrophin. So, for instance, a single gene can be transferred efficiently rather than two or more genes. Therefore, selecting of monogenic disease yields a promising result in this case. Furthermore, the size of the gene also plays an important role in success of the experiment. The gene length is very long. It is very difficult to incorporate and transfer the gene through the vector into the body of the person who is having the genetic disorder. We know very well the uh, dystrophin gene is very high lengthy in nature. So the gene of interest must have the following characteristics. They must have high AT rich sequences, then GC content must be less than 50%. The gene should not contain large number of exons. These are all the characteristics that are defining the case of your gene of interest. Selection of the plasmid. The plasmid plays an important role in delivering the gene of interest to the specific location in the host genome. The plasmid selected for the experiment must be having a high transgene carrying capacity. So the size of the kilobase pair which is carried by the plasmid should be very high in nature. So some of the important sequence that are needed to be inserted along with our DNA. We need a promoter sequence to be added in the 5' end of the DNA of gene of interest. Then we should have inverted terminal repeats for the recognition of the target site in the genome, and we have to have one antibiotic resistant gene and control elements and the gene which are essential for the viral enzymes. Selection of the vector. The vector selection is very important factor in gene therapy. The vector selection process is based on the gene what we are going to insert. Although the viral vector gives an excellent delivery rate and integration deficiency, it is not so safe. Furthermore, the selection of the vector is also based on the size of the gene of interest. For instance, if the size of the transgene is greater than 35 kilobase pairs, the adenovirus should not be recommended because its carrying capacity is lower than this. Selection method. After selecting both the gene of interest as well as the vector, we should decide the method of gene transfer. The target tissue is a somatic cell. The somatic cell gene transfer therapy can be chosen. Choose, choosing ex vivo or in vivo, it depends upon the transgene in which we want to insert. For example, if the transgene for cystic fibrosis, the in vivo gene therapy method work excellently. However, the same method fail in the case of Duchenne muscular dystrophy or AIDS. On the contrary, ex vivo gene therapy gives more precision for disease like hemophilia. So the selection of the method is based on the type of the gene, type of the transgene, type of the tissue, and chance of infection. Generally, ex vivo gene therapy for AIDS does not recommended due to its high risk factor for association when the virus is when this retrovirus is cultured in this case. Selection technique. The fifth step of the gene therapy experiment is selection the technique. If the non-viral vector is selected as we did, The techniques what we can use is sonication, electroporation, magnetoception, gene gun or liposome. The DNA cannot be directly inserted into the host cell, so we need to create pores on the surface of the cell. Electroporation is the best technique for all type of gene transfer experiment with non-viral vectors, even though it works more efficiently along with the liposomes too. Experimental setup. The experimental setup is a crucial step as we see here. First, the gene of interest to be selected. It should be carried out in a highly contaminant-free, sterile and sophisticated setup in the case of the laboratory. The art of the laboratory facilitates gene therapy must be equipped with all the utilities and safe setup. The experiment must be performed under the supervision of the experts. Then only it is successful. After all, the possible applications of gene therapy. The faulty or mutated gene is replaced with healthy gene by using this therapy method. This method can be used for diagnosis of disease, which is approved globally. It is used in the diagnosis of inherited disease such as cystic fibrosis, Duchenne muscular dystrophy, muscular atrophy, and hemophilia. Furthermore, efforts are being made to develop gene therapy against monogenic disorders such as thalassemia and sickle cell anemia. The in vivo gene therapy or viral vector mediated gene therapy is good option for treating diseases like Parkinson's, Alzheimer's, and brain tumors. Nowadays, the gene therapy is open door for dentistry also. It is used to grow teeth, new teeth, bone repair, and teeth generation. DNA vaccines are another futuristic application of gene therapy. The naked DNA is directly injected into the therapeutic along with for the production of therapeutic proteins such as insulin and thrombotic factors. Treating cancer using transgene is another utilization of gene therapy in which the expression of the oncogene is suppressed by a transgene. Also, the infected cancerous cells are likely to be killed by the expressing toxic gene into those cells. Consequently, it promotes apoptosis of the infected cells, or what we call it as suicidal gene therapy. In addition to this condition, cardiac disorders, neurological disorders too are treated by this such technology. Another, one of the first use of ex vivo gene therapy was treating a young girl with a rare genetic disease such as adenosine deaminase deficiency. So, it results in the adenosine deaminase results in the development of severe combined immunodeficiency. We call it as SIDS disease. People with this disorder are missing the adenosine deaminase enzyme, so which affects the immune system. As a result, the individual with ADA deficiency do not have healthy immune system to fight against various infections. To treat this, the doctors took bone marrow cells of the particular baby and they isolated and differentiated all kinds of blood cells from the patient body and added a healthy gene to them in the laboratory, and they put back those healthy genes into the bloodstream. This helped in the patient to immune system to work properly, but the treatment needed to be repeated several times for this case. In the case of patients with macular degeneration, the gene therapy is working out. The retinal RPE65 gene therapy is the breakthrough that pave way for the gene therapy for treating number of retinal diseases, including age-related macular degeneration. We call it as AMD, retinitis pigmentosa, choroideremia, and others. Now that a safe and effective technique has been established for delivering the gene into the retina, additional genes can now be delivered to the retina in the same way and tested for efficacy and safety in clinical trials. The therapeutic strategies for cancer gene therapy: targeting the immune system, targeting the tumor cells. Immunogene therapy: improved tumor recognition by tumor cell gene transfer. Example: cytokines, co-stimulatory molecules, DNA, RNA, and vaccination. T cells redirection via recombinant TCR or CAR gene transfer, APC or T cell activation by gene transfer. Example cytokine tumor neoantigens. Gene correction and editing. Insertion, repair of tumor suppressor or oncogene for growth control. Example P53 RB1 genome. Epigenome editing. Suicidal gene therapy involves transfer of prodrug converting enzymes, that is HSV, TK, CD, or bacterial toxins are injected, thereby it kills the cancerous cell as well as tumor cell. Oncolytic virotherapy. Oncolytic viruses are used for this purpose in treating the cancers. That is example Onyx 015, HSV, NDV, Rio virus and JX 970 are used for this purpose. Gene suppression, that is they prevent the expression of mRNA. Antisense oligonucleotides, decoy ODs, ribozymes, silent RNA and shRNA and miRNA, all these are when they are inserted, they prevents the expression of the particular gene. Thereby, it reduces the size of tumor. It may be normal or a cancerous tumor. Limitations. Even though we have a potential application in the case of gene therapy, but it has different problems that find it very difficult in carrying out in the clinical trials on human. The viral vectors which are used in gene therapy can infect the host cell nearby healthy cells and produce a strong immune response against them. Further, the transgene are not, when they are inserted, they are not always expressed all the times. Due to short-lived nature of the transgene, the success rate of the gene therapy is very low. The gene transfer experiments are restricted only to live human embryos because of the ethical issue which are associated with it, because as if we are playing against the god. Rapid integration of the transgene is not possible, therefore we don't know when will the transgene will be expressing. Further, not all the cells of the particular tissue accepts the transgene. Random integration to the other location can result in adverse effects that a normal gene can be can that a gene can activate pro-oncogene to oncogene, or it may deactivate a normal gene. The cost of the gene therapy is very high. That is a major problem. It costs around $1 million per therapy, where the insurance company fails to cover it. So these are all the limitations of gene therapy. Even though it has major positive points in treatment of various genetic disorders, in future this will further changes will occur in the case of treatment procedures and cost also as the research advances. Thank you.